Hello everyone, my name is Ariel Hill and welcome to my week 1 presentation for Biology 5402, Immunology and Microbiology. This week's focus is on bacterial pathogenesis, how bacteria causes disease, and how the incredible defense mechanisms God designed within the human body. This presentation will give an overview of how bacterial pathogens invade the body, how the immune system fights them, and how antibiotics assist in healing. By the end, you should be able to describe bacterial pathogenesis as well as innate and pharmacological resistance mechanisms. The content comes from Dr. Curtis Henderson's lectures, our textbook by Levinson, and several educational videos. Bacteria are single-celled organisms that can be both helpful and harmful. Beneficial bacteria live in our gut or help produce food, but pathogenic bacteria cause infection by invading tissues or releasing toxins. Examples include Streptococcus pyogenes, Staphylococcus aureus, and certain E. coli strains. Pathogenesis means the process by which bacteria cause disease. To succeed, bacteria must first enter the host, attach to cells, invade tissues, avoid immune detection, cause damage, and eventually exit to infect others. Each stage provides insight into how we can prevent or treat bacterial infections. Infection occurs in 4 predictable stages. During incubation, bacteria multiply quietly without symptoms. The prodromal stage brings mild signs such as fatigue or fever. In invasion, symptoms peak as the pathogen spreads. Finally, during convalescence, the body recovers as immunity clears the infection. Several bacterial features determine how severe an infection becomes. Adhesions allow bacteria to stick to host cells. Toxins damage tissues and disrupt cell functions. Invasions help bacteria move through tissue barriers, and immune evasion strategies like capsules or biofilms help them hide from white blood cells. Let's look at a few examples. Staphylococcus aureus causes boils, wound infections, and sometimes sepsis. Streptococcus pyogenes is responsible for strep throat and scarlet fever. Mycobacterium tuberculosis causes tuberculosis by surviving inside immune cells for years. These examples show how versatile and adaptive bacteria can be. The body's innate defenses act immediately and non-specifically. Our skin, mucous membranes, and stomach acid form the first line of defense. Inside the body, white blood cells like neutrophils and macrophages engulf bacteria, while inflammation and fever create hostile conditions for pathogens. Unlike the innate system, the adaptive immune response is slower but precise. B cells produce antibodies that recognize and neutralize bacteria, while T cells destroy infected cells and assist other immune functions. After infection, memory cells remain, providing long-term protection, the principle behind vaccines. Antibiotics target vital bacterial processes such as cell wall formation or protein synthesis. However, misuse and overuse of antibiotics allow resistant strains like MRSA to emerge. That's why healthcare professionals must use antibiotics responsibly and support research for new antimicrobial therapies. Dr. Henderson reminds us that studying microbiology deepens our appreciation for God's creation. Every cell and immune response reflects His design and purpose. Psalm 139 verse 14 says, "I praise you because I am fearfully and wonderfully made." As Christians in science, we are called to use knowledge ethically and compassionately to promote healing. To summarize, bacterial pathogenesis follows predictable stages, but our immune system and antibiotics work together to defend us. Week 1 emphasized that understanding microbes helps us prevent disease and promote health. When combined with faith, this knowledge reminds us that scientific work is a way to glorify God through service to others. These references list the course readings, YouTube lectures, and textbook chapters used in preparing this presentation, all cited in APA 7th edition format.